Hello. Good day, everyone. Welcome to Parasitology Part 2. Here, we will discuss the part of parasitology as well as the different parasites and their life cycle. In this chapter, we provide a summary of the life cycles and biology of some of the most important parasitic protozoa in human and veterinary medicine. We also briefly mention fungi and plants, but there is insufficient space to deal with these topics in any depth. Parasitic protozoa can be found living in all the organs of the body and cause diseases that range from benign to rapidly and incurably fatal. They also exhibit very imaginable means of transmission from contamination to sexual and vector-assisted. The parasitic protozoa. The kingdom Protista is a loose assemblage of organisms that have in common their shared characteristic of possessing a membrane-bound nucleus, which therefore makes them eukaryotes, and their lack of the organizational features that are found in the kingdoms Fungi, Plantae, and Animalia. Although they are for the most part single-celled, some are colonial, and in the case of the brown seaweeds and kelp, these can form structures that extend to several meters in size. Under this kingdom is the phylum Rhizopoda. This phylum, which used to be known as Sarcomastigophora, contains the amoebas. The name Rhizopoda translates as root-like foot and refers to the process by which the cytoplasm flows within the cell to cause projections of the body wall called pseudopodia or false feet that are used for both movement and the ingestion of food. Food is ingested in a process called phagocytosis and results in the food item being enclosed within a membrane-bound vesicle called a food vacuole. In some species, movement is also aided by one or more flagellae. Although the amoebas are sometimes said to be primitive, ultrastructural and molecular studies indicate that this is not the case. It is a relatively small phylum consisting of about 200 described species, most of which are free-living or commensal, but the phylum includes important parasitic species such as Entamoeba histolytica. Under phylum Rhizopoda is genus Entamoeba. One of the species of genus Entamoeba is Entamoeba histolytica. This is a causative agent of amoebic dysentery. Dysentery is a general term that is used to describe a serious inflammatory disorder affecting the intestines that results in intense diarrhea and is often accompanied by pain and fever. It can result from a variety of causes and amoebic and bacterial dysentery which occurs in both temperate and tropical regions. Dysentery has long been known as handmaiden of war, often inflicting more casualties than bullets and bombs. Accounts of epidemics of dysentery accompany nearly every account of war from antiquity to the present day. Wherever large numbers of people, especially if they are malnourished, live in close proximity and squalid conditions, the situation is ripe for an outbreak of dysentery of epidemic proportions. Not surprisingly, the increased migrations and deteriorating economic conditions in many developing countries have led to enhanced levels of amoebic dysentery in recent years. Tourists traveling on exotic holidays have also found themselves victims of amoebic dysentery. This is a picture of Entamoeba histolytica. There are two stages in the life cycle of Entamoeba histolytica. The actively growing and feeding stage, referred to as the trophozoite form, and the transmission stage, called the cyst form. Like all other parasitic protozoa, but unlike the free-living forms, Entamoeba histolytica has no contractile vacuole. It also lacks mitochondria. This figure shows the Entamoeba histolytica trophozoite and the cyst form. In the amoeba, the spore is morphologically indistinguishable from Entamoeba histolytica and has the same life cycle, but it is non-pathogenic. It is much more prevalent than Entamoeba histolytica, and therefore it is essential to be able to distinguish between the two species in order to avoid a false diagnosis of amoebic dysentery, and thereby initiating inappropriate treatment. Entamoeba moshkovski, for many years, this species was considered to be a free-living amoeba. It was originally described from Moscow sewage and subsequently identified from a variety of ponds and sediments around the world. Entamoeba gingivalis, this amoeba is commonly found in swab samples taken from the gingival crevices of the human mouth. Entamoeba gingivalis does not form cysts, and therefore transmission is probably through kissing or sharing food and eating implements, although it is often considered to be involved in the development of periodontitis, an inflammatory disease that affects the gums and the bones surrounding the teeth. It is found in both healthy and diseased individuals. Like the other members of the genus, Naegleria fowleri is a free-living amoeba flagellate. It is an amoeba which, in one of its life cycle stages, possesses flagellae. It is a cosmopolitan species that is normally found in freshwater ponds and lakes, but it has also been recovered from a wide range of wet or moist environments, such as swimming pools, humidifier systems, and damp soil. Balamuthia mandriliaris. This is another cosmopolitan amoeba that is capable of causing fatal encephalitis as an apparently opportunistic infection. Its name is derived from it being discovered as the cause of a fatal brain infection of a mandrill baboon that died at San Diego Wild Animal Park in California in 1986. Acanthamoeba. The genus Acanthamoeba contains over 20 species, and they are among the most common free-living soil amoebae. They are also found in a variety of freshwater habitats, and several have been recorded as opportunistic pathogens. One phylum which belongs to kingdom Protista is Phylum Metamonada. Belonging to this phylum is the order Trichomonadita. Most protozoa belonging to this order are parasites, the best known of which is the genus Giardia, which is shown in the picture. They are characterized by the presence of two haploid nuclei, each of which is associated with four basal bodies and flagellae. They lack Golgi apparatus and mitochondria, but organelles called mitosomes that are thought to represent relic mitochondria have been identified in some species. One species of the genus Giardia is Giardia intestinalis. This causes an infectious disease, Giardiasis. The cyst form is passed through the contamination of water, food, or hands or fomites with infective cyst. This enters through the mouth by eating or drinking and enters the intestine. In the intestine, the cyst form is transformed to a trophozoid diagnostic stage. This trophozoid form will reproduce, and the adult trophozoid will produce the cyst form. Trophozoites are also passed in stool, but they do not survive in the environment. Another order belonging to phylum Metamonada is order Trichomonadita. Under this order is Histomonas miliagradis. This is an extremely common parasite of humans, and despite its name, it is frequently found in men, though it tends to be less harmful in them. The parasite is teardrop-shaped, with 5 flagella emerging at the anterior end. 4 of these flagella are free, while the fifth curves back to form a short undulating membrane that extends just over half the length of the cell. Only one body shape. This parasite infects a wide range of birds, but for some reason it is particularly pathogenic in young turkeys, in which untreated infections are usually fatal. It is found within the lumen of the cecum and the liver parenchyma and gives rise to the disease histomoniasis. Another member of order Trichomonadita is the Trichomonas vaginalis. Although millions of women are infected with Trichomonas vaginalis every year, in the majority of cases, these are asymptomatic. The protozoa feed on bacteria and slough off epithelial cells present in the reproductive tract. However, in some cases the parasites cause a severe inflammatory response that is manifested as a copious frothy white or greenish vaginal discharge. There appears to be interaction between the protozoan and other sexually transmitted organisms. In particular, Trichomonas vaginalis is thought to enhance the transmission of HIV is expressed. Although the size can vary considerably, the length can be 7 to 32 meters, while the width is 5 to 12 meters. Another phylum belonging to kingdom Protista is Phylum Epicomplexa. This is one of the largest of the protozoa phyla and includes species that are important parasites of humans and domestic animals. All members of the phylum are obligate intracellular parasites of invertebrates and vertebrates. A common feature shared by all apicomplexans is the presence within their invasive stage of a unique intracellular structure called the apical complex that is composed of a group of secretory organelles called the micronyms and rough trees. The complex is situated at the anterior apex of the cell, where it is associated with a region called the oral structure. The genus Plasmodium is believed to have evolved hundreds of millions of years ago, and long before the arrival of the vertebrates. Over 200 species have been described, most of which are parasites of birds, although there are also many that infect reptiles, rodents, and primates, and they are transmitted by blood-feeding invertebrates, especially mosquitoes. There are 4 principal species that infect humans: Plasmodium falciparum, Plasmodium vivax, Plasmodium ovale, and Plasmodium malariae. There is a 5th species, Plasmodium knowlesi, which is normally considered to be a parasite of monkeys that can cause fatal infections in humans and certain other species, which are usually parasites of other animals that cause occasional infections. In most vertebrate hosts, Plasmodium parasites are not particularly pathogenic, but in humans they can cause the disease malaria. Plasmodium falciparum. This causes the most dangerous form of malaria, as it can develop into the potentially fatal cerebral malaria, also sometimes referred to as malignant tertian malaria. Its distribution has become restricted in recent years, although it is still very common in tropical and subtropical regions such as sub-Saharan Africa, Southeast Asia, and parts of South America. It is the main cause of the mortality associated with malaria. It does not form hypnozoites, but sudden relapses can occur months or even a year or more after the last fever episode as a result of the parasite persisting in the blood at low subclinical levels. The merozoites are able to invade and develop in both young red blood cells and mature red blood cells. Consequently, Plasmodium falciparum malaria is often characterized by high levels of parasitemia. The malaria parasite life cycle involves two hosts. During a blood meal, a malaria-infected female Anopheles mosquito inoculates sporozoite into the human host. Sporozoites infect liver cells called hepatocyte and form into schizont. This schizont releases merozoite. A dormant stage, hypnozoites can persist in the liver if untreated and cause relapse by invading the bloodstream, maybe in weeks or even years later. After this initial replication in the liver, the parasites undergo asexual reproduction. Merozoites infect the red blood cells. The ring stage, in which trophozoites transforms into schizont. This schizont then rupture, releasing the merozoite in the bloodstream. Blood stage parasites are responsible for the clinical manifestations of the disease. The gametocytes, male microgametocytes, and the female macrogametocytes are ingested by the Anopheles mosquito during a blood meal. The parasite's multiplication in the mosquito is known as the sporogonic cycle, while in the mosquito's stomach, the microgamete penetrates the macrogamete, forming into a zygote. The zygotes, in turn, become motile and elongated, which is known the ookinete. These invade the midgut wall of the mosquito, where they develop into oocysts. The oocysts grow, rupture, and release sporozoites, which make their way to the mosquito's salivary glands. Inoculation of these sporozoites into a new human host perpetuates the malaria life cycle. Another phylum belonging to kingdom Protista is the phylum Epicomplexa. Under this phylum is the genus Toxoplasma, which contains just a single species, called the Toxoplasma gondii, but what it lacks in member species, it makes up for in numbers of hosts, since it is probably capable of infecting all mammals and birds. Toxoplasma gondii. This intracellular parasite was initially described from a desert rodent, the North African gondii. The name Toxoplasma has nothing to do with the toxins, but it is derived from the curved shape of the tachyzoid stage of the life cycle. Toxon is the Greek word for a bow or something that is crescent-shaped, and plasma is Greek for creature. The life cycle of Toxoplasma gondii has two parts and three infectious stages. The two parts of the life cycle are the sexual cycle that occurs within cats and other felines, which are the definitive host, and the asexual cycle that occurs in the intermediate host, which are virtually any warm-blooded animal, including humans. The three infectious stages are the sporulated oocysts that contain the sporozoites, the tachyzoids, and the tissue cysts that contain bradyzoids. All three stages are infectious to both the feline definitive host and the intermediate host. Cats acquire their infection by consuming either sporulated oocysts passed in another cat's feces, or through contamination or an intermediate host containing the tachyzoids and bradyzoids. Following ingestion, the parasites invade the epithelial cells. This one. These epithelial cells makes up the lining of the chiasmal endocyne and undergo a complex series of asexual divisions. The first step is multiple or multiplication by endodyogeny. This is an unusual form of division in which two daughter cells develop within a mother cell, which is then consumed by her offspring before separation takes place. Endogyogeny is followed by endopolygyny, in which the mother cell produces several daughter cells simultaneously. This is then followed by gametogenesis. Then there is a formation of microgametes and macrogametes. After fusion of the gametes, a zygote is formed, and this develops into an oocyst that is shed with the feces while it is still unsporulated. If the cyst is ingested by a new host, at this stage it is not infectious. Sporogony therefore happens outside the host and takes about 2 to 3 days under ideal conditions. The infectious sporulated oocysts are oval in shape and contain two sporocysts, each of which contains 4 sporozoites. The sporocysts can remain infectious in the environment for months and possibly several years. The shedding of oocysts starts 3 to 10 days after initial infection and continues for only 1 to 2 weeks, although some estimates suggest that a cat may shed up to 100 million oocysts in its feces during that time. After an initial infection, a cat is usually resistant to reinfection. The prolonged survival of the oocysts in the environment compensates for the short period over which they are shed. In some cats, developmental stages may persist in a dormant state and give rise to another batch of oocysts at a later date following their reactivation. This could happen as a result of a change in the host's immune status following challenge by another infectious disease. Alright, well, I guess that's all for the part 2 of our parasitology session. Thank you and have a great day. See you in the next one. Bye.